I'm Smriga.
And I'm Vicente Fernandez. We're both postdocs in Roman Stalker's lab in the Civil and Environmental Engineering department.
And what we study in our lab is interactions between microbes that live in the ocean. So in the image, what you're looking at is a diatom, and diatoms are an example of phytoplankton, and phytoplankton are microalgae that are the primary producers for the most part in the ocean. The diatom that you're looking at here is called a Catastris species, and the main part of the diatom is the rectangular portion that's in the middle, and each of these individuals that are in this diatom chain have spines. In the blue in this image, those are individual bacteria cells, and bacteria are the most numerous organisms in the ocean. They're sort of like the decomposers, and so what you're looking at in this image is phytoplankton that might be on its way towards death. And here bacteria are picking up chemical cues that are coming out of this diatom, and they're in fact helping to decompose and recycle the carbon and nutrients that are associated with this phytoplankton cell.
In order to be able to find the resources in this difficult, small, low Reynolds number environment, bacteria do something called chemotaxis, in which they're able to, over time, move in the direction upwards through chemical gradients in the direction of more nutrients. And the way they do that is that most of the motion is kind of a random walk of various sorts for bacteria, but they're able to bias this random walk by changing at what points they change direction. Even though they don't have control over the specific direction, by controlling when they change direction, they're able to continuously move on average towards nutrient sources. And you can see here that the bacteria are definitely accumulating and swarming near the diatom. But if you look carefully at the individual tracks, you can see that the bacteria don't move in a particularly conscious and directed way. They're not all moving towards the diatom. And so in our research, we've been able to investigate how different levels of diatom availability, such as low levels of kind of normal seawater, or during a diatom bloom or during a diatom bloom collapse, how that will affect both the distribution of motile versus non-motile bacteria and the overall quantity of bacteria.
Bacteria-phytoplankton interactions are, we think, fundamental to the way that carbon and nutrients are cycled in the ocean.
Given how vast the oceans are, I find it really fascinating that such an important important facet of the interaction between the life in the ocean is at such a small scale. I mean, a couple hundred microns, and yet this is sort of a key interaction in those food webs. So I find it's really exciting that we've been able to start to quantify and look at and in detail analyze some of these interactions. Although this is our image in our work currently is about one specific diatom and a natural population of bacteria, it's really sort of this prototypical interaction between a sudden food resource and general bacteria. So it's super exciting in kind of the broad applicability to microscale interactions of microbes in the ocean. And that shift from these huge, nearly global scale processes to this minuscule 100 micron interaction is really exciting to me.